Hello everyone, welcome to the Crazy Science. আমি তোমাদের biology tutor. I am present here again with another topic, and that topic is the third part of molecular basis of inheritance. So molecular basis of inheritance আজকে আমি শুরু করব from the transcription. In the last class we talked about the translation process. যেখানে Watson and Crick they talked about the leading and lagging strand. ও জায়গাকে ফ্রেগমেন্ট আমরা পড়েছি, and in this part আমরা পড়ব the transcription, translation, template, codon. তারপরে ট্রান্সক্রিপশনের দুটো পার্টস থাকে, one is a prokaryotic and there is a eukaryotic. And other than this translation, amino acids কিভাবে ফর্ম হয়, জেনেটিক কোডনটা কি, and এরপরে ভিডিওতে এটার পরে যে ভিডিওটা বানাবো that would be the last video of the molecular basis of inheritance. তো ওটাতে জেনেটিক কোডন মিউটেশন ব্যাপারে আরো আমরা ডিটেইলসে আলোচনা করেছি, বাট ইন দিস ওয়ান উইল টক অ্যাবাউট দ্য ট্রান্সক্রিপশন এন্ড ট্রান্সলেশন, which is very important, and in every alternative year it comes. So let's begin today's class. So as you can see, I have given the heading, that is transcription. So transcription maneta ki? This transcription maneta holo, it is something process. It ekta process jekhane ki form hoche RNA is forming. Here RNA is forming in this transcriptional part, and in this one, so in the transcriptional part we'll be talking about the forming of what? Forming of this RNA from the DNA itself. And translation আমরা কি দেখেছি? In the translation আমরা দেখেছি how DNA is forming, where leading and lagging strand was there. Okay, so let's start. So before starting it, আমরা just বলতে চাই যে central dogma I also talked about. The transcription means I'm talking about that the formation of the RNA molecule. So first it started from the DNA. So firstly it started from the DNA itself. So এটা DNA থেকে শুরু হয়েছে, DNA, and where the replication এ আমরা কি দেখেছি? Leading and lagging strand, where we got to see, and later it got joined by the definitely DNA ligase enzyme itself. তো একটা ছিল লিডিং স্ট্র্যান্ড আর একটা ছিল ল্যাগিং স্ট্র্যান্ড। লিডিং টেন্স which is very continuous, এটা পড়েছি লিডিং, one was a leading strand, another was a lagging strand. So leading strand was a continuous one, and whereas the lagging strand was a discontinuous one. Okay, discontinuous one. So basically DNA first of all form hobe, the DNA form wata ba or the replication what the formula ta ba process take, it is called the replication itself. From the DNA RNA will be formed, from the RNA ultimately protein will be formed. All right, so DNA to RNA process টাকে বলা হয় that is replication, transcription. It is called the transcription. It is called the transcription. And from the RNA to protein forming, the journey is called the translation. And where the DNA is getting formed from the discontinuous and continuously strand is forming, this type of formation of the DNA strand is called the replication. Okay, so replication, transcription and the translation, these are the few things. Okay, all right. So leading strand, one was leading, another is lagging. Now we'll be talking about the transcription process. Before jumping into the eukaryotic and prokaryotic, we need to know basic idea about the transcription. So let's begin from here. As I prepared the slide, it is the process of the copying genetic information from one strand of the DNA into RNA. So basically our main motto is to make the RNA. And the form of making RNA from where? From the DNA itself. তো এইভাবে যে প্রসেসটাকে আমরা কি বলি, এটাকে বলে ট্রান্সক্রিপশন। and in the transcription only one segment of the DNA and only one strand is copied in RNA. তো বেসিক্যালি একটা স্ট্র্যান্ড, ডিএনএ তো দুটো স্ট্র্যান্ড থাকে না, তো একটা স্ট্র্যান্ড এখানে কপি হবে, দ্যাট ইজ অনলি ওয়ান স্ট্র্যান্ড উইল বি কপিড হিয়ার। The adenosine forms base pair with the uracil instead of the thymine. I already told you in the DNA you will get to know about the ATGC, right? Adenine, thymine, guanine, cytosine. But in the RNA, the adenine, uracil, guanosine and cytosine will be there. Instead of thymine, the uracil will be there, and uracil is very reactive, very destructible, easily broken and easily react very much, whereas the DNA in the DNA thymine is very less reactive, very stable. So for that reason they are present in DNA and they give more stability to the body, and two, that's why DNA carries the genetic information. There is no one other formula. DNA is quite powerful just because they have the thymine in their base pair, as their base pair, nitrogenous pair. Okay, and uracil, they have the uracil, means in the RNA itself. The transcription of the DNA, ট্রান্সক্রিপশন ডিএনএ মতলব হলো যে তারা আরএনএ ফর্ম করছে, তারা প্রোটিন ফর্ম করবে ইন দ্য ফিউচার উইথ দ্য হেল্প অফ দ্য translation process. Includes the promoter, which is we call P in short. Next is a structural gene and the terminator. Okay, so three components you require if you are doing the transcription over here. So amra dekhbo in the next slide we'll get to see the transcription. We will get to see the in two parts it will happen. One is a eukaryotes and other is a prokaryotes. Prokaryotes, we all know that bacteria, and the eukaryotes means that advanced persons, advanced animal, advanced organism, those who have a proper nucleus. Okay, all right. So these are the three parameters. One is a promoter, the first one, the second one is a structural gene, and the third one is a terminator. All right, the strands that has the polarity 3' to 5' act as a template, and it is called the template strand, whereas the strand is called the coding strand. Other means ota 5' থেকে 3' এ হবে. This is called the coding strand. So as you can see, this diagram is also in the NCERT. So nothing is out of NCERT, everything is in the NCERT. I have taken all the information from the NCERT itself, and you can see there is a 3' to 5', another is a 5' to 3'. Okay, so those are the 5' to 3', they are the coding strand, and those are the 3' to 5', these are called the what? Template strand. These are the template strand. ঠিক আছে, তো একটা দেখছো যে 5' থেকে 3' এ চলে যাচ্ছে, এটা হলো কোডিং স্ট্র্যান্ড, আর একটা উল্টো 5' থেকে 3' to 5', they are going and they are totally opposite, alternate to each other, that is called the template one. And the promoter means the সামনে যে জায়গাটা যে থাকে, সামনে যে স্ট্র্যান্ডের যে একদম ফ্রন্টাল যে জায়গাটা থাকে, this is called the promoter. ওখান থেকে কি হয়, ট্রান্সক্রিপশন জাস্ট স্টার্ট হয়, ওখান থেকে বেসিসটা শুরু হয়। ট্রান্সক্রিপশনের যে প্রসেসটা থাকে মিনস ফ্রম ফর্মিং দ্য আরএনএ, দ্য জার্নি বিগিনস ফ্রম দেয়ার। ঠিক আছে, and this entire is a structure, the middle part is a structural gene, and the template strand are last terminator, where they cannot able to do any further transcription or further RNA process, যে RNA বানানোর যে প্রসেসটা কন্টিনিউ করতে পারে না, and that time they just terminate that, because RNA is just a single strand, it's just a single strand, this is nothing more that, but DNA itself a double stranded, and this is what we read in the last video, that replication what's in Greek semi-conservative বলেছে, because already we have the certain kind of idea about the DNA itself. ঠিক আছে, semi-conservative it is called. So one is a lagging, another is a leading, means continuous and discontinuous. There are also the 5' to 3', 3' to 5' continuous. All right, okay. So move to the next slide. This was the basics and with the diagram. So here is a little bit differences. You might have certain kind of confusion. What is leading? What is lagging? Why the 5' to 3', the 3' to 5', they are totally opposite to each other. There's certain kinds of একটা এখানে অ্যাক্টিভ থাকবে আর একটা এখানে অ্যাক্টিভ থাকবে না, মতলবটা এটাই, কারণ দুটো একসাথে they cannot able to work properly, it's not possible. Okay, all right. So this is a template strand, as you can see. So the template strand, another is a coding strand. So it is a DNA strand with the 3' to 5' polarity. As I said earlier also, that two will be parallel to each other and they will never intersect. There are two strands, so you can see this for a better understanding. I'm telling you, এভাবে draw করে তোমাকে দেখানো হচ্ছে. One is a 3' to 5' polarity and coding strand is DNA strand 5' to 3' polarity. Means I can write here 5' to 3', and there is a 3' to 5'. All right. So this upper one is a template one, and this lower part is a coding one. All right, but their way of direction is totally different. Polarity is different. All right. Act as a template for the transcription and codes for RNA. They act as a template. They are the ultimatum who can make the RNA itself, where it does not code for any region of RNA during the transcription. So basically, এখানে দুটো মতলব এটাই হলো, এখানে পুরোপুরি template is working, but coding is not working. Code is totally useless. Code is not coding anything. They are not forming RNA. Coding ultimately not forming RNA. Your main motto in the transcription is to make the RNA. Means a single-stranded, right? Single-stranded genetic material. Itself a material, but it does not code anything. But RNA is something that they are actively participate in the protein synthesis. But DNA is something that which carries the information. But both are the genetic material here. All right. But template ta ki korche? They code for the RNA. All right, so this is a template one, this is a coding one, this is a template coding. So basically template is more useful than the coding one. Coding does not code anything, and this just during the transcription. And transcription এর সময় যে কোডিং করছে, যে properly RNA forming করছে, তারা active থাকবেন. They are the ultimatum, and two strands cannot act at the same time properly. So one will work, another will be in a pause. All right. So this difference make asked. It's very important. Moving to the next slide. Transcription. Now we have reached in the process stages itself. So in stage আমরা দেখব কি? In this stage I'll be talking about the, as I said, এতক্ষণ আমরা কি পড়েছি, little bit ideas. We took the idea. Now transcription is a process porbo. Stages porbo. Stages. There are the three stages. Okay. One is initiation, where the process of making RNA will be start. Next one is a elongation. Elongation means whatever you're making, it will be elongated. Third one is termination. Means now your process is already done. That RNA already produced from the DNA itself, it will be separated and it will go it will go in another region for further process, protein synthesis. So we'll start the initiation with it. This is a Roman 1. RNA polymerase attaches to the DNA molecule and moves along with the DNA strand until it recognizes the promoter sequence. So first of all, if this is your DNA, so suppose this is your DNA, right? So in this DNA, what happened? RNA polymerase, they just move around, ঘুরতে থাকে, just to find the tumor promoter region konta. Promoter means this is the initiation point from where the synthesis of making RNA বা RNA বানানোর যে প্রসেসটা এটা স্টার্ট হয়ে যাবে. ঠিক আছে? তো যে জাস্ট RNA polymerase যে এনজাইমসটা থাকে, যেটা ঘুরতে থাকে, just they give a make around. যে ঘুরতে থাকে কোথায় এটা, where they will get to see their proper place. কোথায় তারা proper place পাবে বসতে. Okay. They are known the transcription start site. The DNA double helix that unwinds all the bases of the DNA strand are exposed. তারা আস্তে আস্তে খুলতে থাকে. They are look like this না, এইভাবে তাদের they're having the nitrogenous base, but when they make the protein, RNA, they just unwind themselves. It আস্তে আস্তে কি করতে থাকে, এটা আস্তে আস্তে খুলতে থাকে. Suppose এভাবে খুলে যাবে, Okay. This will be more broader. জায়গাটা আরো broad হয়ে যাবে. This will be more broader. The DNA molecule will be more broader. Okay, so it will be more broader like this. It will unwind হয়ে যাবে. Nitrogenous base will be like this. There will be little bit gap in between the two stranded. Okay, all right. And this acts as a template for the new mRNA strand. Template means they will start the initiation. They will participate in the RNA making process. All right. Next, your next part is elongation. First of all, keep on. RNA polymerase will be there. They will try to find the region where they will start their promoter region, where they start forming. কোথায় তার দেখতে খুঁজতে শুরু করবে, কোথায় তাদের promoter region, যে জায়গাটা পেয়ে নিল, the DNA will be start opening themselves. Next, elongation. Elongation, ribonucleotides are added to the template strand and enables the growth of the mRNA. And mRNA যে গ্রোথটা ওটাকে আরো বেশি বাড়িয়ে দেয়, এনহ্যান্স করে দেয়. All right, and termination টা কি হবে? Termination হবে now you already entered, you elongated করেছো, বানিয়েছো RNA. Now it's time to leave the stage. Means that this is the RNA already formed that will leave the process, and the new RNA process বা আরেকটা নতুন RNA বানানোর যে স্টেজটা বা তাদের যে জার্নিটা, it will start. Okay, the RNA polymerase encounters the terminate sequence and the transcription will be stopped, and the RNA, suppose এখান থেকে একটা RNA formed হলো, তো এটা লেগেছিল এটার সাথে. In the next process, you see that RNA will be separated when the termination or terminator codon or the factor will be attached to it. RNA polymerase then releases the DNA template. So automatically যেটা unwind হয়েছিল, it will be again close, and they will wind together. They will be just আস্তে আস্তে ওরা নিজেকে পেঁচিয়ে নেবে, ওরা যেটা automatically close হয়ে যাবে. So basically this is very simple, and প্রত্যেকটা প্রসেসে তোমরা এরকম দেখতে পারবে. তো So I was talking about this process, transcription process, যেখানে আমরা already I said to you that transcription is quite simple, and শুধু একটু differentiation দেখতে পারবে eukaryotic and prokaryotic মাঝে. So transcription means where you are making the RNA. You will make the RNA, right? The transcription basically two types থাকে. One is a prokaryotic, another is a eukaryotic. So whatever I said in the initiation process, whatever said in the elongation process, where that RNA will be elongated and will be matured enough to separate, and whatever is in the termination process. Termination means it will be terminated, the process will be terminated, and the old, whatever the DNA they make, RNA they make, it will be separated. The new RNA formation journey will be started. So it not very close, very common, but there is a little difference in between the prokaryotes and eukaryotes. As you can see the diagram over here, this is a promoter. Okay, the promoter region is from where the initiation starts. So this is RNA polymerase. You require a factor, and that factor is called the sigma factor, and that factor will start the promoter, that factor will attach with the promoter region, and this factor is not alone. This factor is connected with the RNA polymerase, where they are getting the energy and also support to find the promoter region to start the transcription process. And this is how they will start. You can see they are getting unwind. আস্তে আস্তে খুলে যাচ্ছে, যেটা পেঁচিয়ে ছিল একসাথে এটা আস্তে আস্তে খুলে যাচ্ছে. They are getting more broader, right? আস্তে আস্তে বড় হয়ে যাচ্ছে, and this is nothing but this is our RNA polymerase. You can see the RNA polymerase inside, and the rho sigma factor is there. ঠিক আছে? তো এভাবে তারা RNA আস্তে আস্তে বানাচ্ছে. You can see the RNA they are making here. Okay, তারা বানাচ্ছে on the way. Elongation means they are making it. Okay, elongation means they are making it. So 3' to 5', 5' to 3'2 strand already পড়েছি. One is a coding and it is template, and the entire thing is happening in the template one, not in coding. All right, in the termination process you can see that this is the last phase তারা পৌঁছে গেছে, and where the RNA polymerase is not connected with the sigma now. Sigma was there, sigma had the touch with that, but elongation sigma already separated. Only RNA polymerase was there. But in the termination you can see that rho factor is attached. Another factor came, and that factor is a terminating factor, and that factor will connect with this one, and যে moment they will connect the rho factor, ঠিক আছে, the rho factor when it will connect, RNA automatically will be separated and the new RNA will form, and this is basically diagram টা হলো prokaryotes, means bacterial transcription. This is a bacterial transcription. Okay, so maximally they are close to each other, but there is a little capping and tailing to add hoy in the eukaryotes. Okay, so I want to show you the eukaryotes one as well. তো ওটা দেখলে আরেকটু, এটা হলো আমাদের পিকেটা যেটা আমি বললাম, সিগমা রো। ওকে, সিগমা রো দেয়ার, অ্যান্ড ইউক্যারিওটসের এটার মধ্যে সিগমা রো থাকে, সেমই থাকে, বাট ক্যাপিং, টেলিং and splicing, it's just adding one. So o jagata most of them are common, but there are 2 points you have to add it. Okay, all right. Capping, so it's just for the EK, it's just for the eukaryotes, it's just for the eukaryotes. Okay, the advance, but in the PK you pick prokaryotes, capping, tailing, splicing, they are not there. Okay, তারা বহুত simple থাকে, process is same but little bit একটু add up in the middle, ঠিক আছে, elongation process you can say. In the capping, the methylated guanine cap is added to the protect the mRNA. So basically they are the mRNA. Okay, you can see they are the mRNA. mRNA means the messenger RNA, and involves additional methylated guanine. It occurs 5' of the mRNA transcript. It protects the mRNA from the degradation. Degradation means bhangte. mRNA ekta smooth thake, erokom thake, এটা তো ভেঙে না যায় like this. Okay, next is, this is just a capping. It's only present, these 3 processes only present in the eukaryotes. Next is polyadenylation. Polyadylation means it will be connected with the tail part. Okay, it also protects the mRNA from the degradation. Means main kaaj holo we have to secure the mRNA. mRNA should not be degraded by any means. It involves the endonuclease cleave the mRNA at a specific sequence. The enzyme polymerase facilitates the addition of the severe adenine nucleotides. Next is splicing. Splicing means a non-coding sequence, that is the introns are removed by the spliceosome excision. The coding sequence or the exon joined together by the ligation. Ligation means when they are connected to each other. If they break হয়ে যায়, they will be connected because certain kind of enzymes are there. তো ওখানে একটা কানেক্ট হয়ে যায়। এখান থেকে দেখতে পাচ্ছ ক্যাপিং মিনস দে আর হ্যাভিং এ সার্টেন কাইন্ড অফ সাপোর্ট, জাস্ট ডিগ্রেডেশন থেকে বাঁচানোর জন্য। সো হিয়ার ইজ এ ক্যাপিং, এখানে লাগছে গুয়ানোসিনের, আর দেখতে পাচ্ছ পিছন দিকে টেলিং হচ্ছে, এর পিছনে জায়গাতে টেলিং হচ্ছে পলিএটেইল, দ্যাট ইজ পলিএডিনাইল, যে পলিএডিনাইলের এখানে টেলিং হচ্ছে সেম ডিগ্রেডেশন থেকে বাঁচানোর জন্য। অ্যান্ড স্প্লাইসিং মিনস দে আর জাস্ট few parts of that, means এই যে পার্টটা তুমি দেখছো, few parts of the exon, few parts of the intron. This is a intron, this is a intron, this is a intron, this is a intron, this is a intron. At the end of the day কি হচ্ছে, spliceosome, there is a certain kind of enzyme, with the help of the spliceosome এই যে introns গুলি যেগুলো আছে, introns গুলো they are getting separated like this, they are getting separated, and spliceosome enzyme এসে এভাবে তারা লাগেজ করে দিচ্ছে, they are just connecting the adding means degrading part, and এভাবে তারা mRNA smooth mRNA বানাচ্ছে। So in the eukaryotes it's not easy to make a smooth mRNA, so we have to go through the capping, tailing and the splicing process. Only then you will get a smooth mRNA, because it has to go through a lot of phases, and eukaryotes is advanced animal, it is present, so it's not simple like as the prokaryotes. All right, moving to the next one. So RNA আমরা জানি, RNA is hnRNA, I will say hnRNA যে জিনিসটা থাকে, আমি আজকে একটু RNA ব্যাপারে বলবো। After the genetic codon, more genetic codon we'll get to see in the next video. Adaptive molecules and the translation. So translation is very important, but before that, what is RNA, you have to know that as well. So the RNA means we all know that ribonucleic acid, and the hnRNA means heterogeneous nuclear RNA. Heterogeneous, there are a lot of spaces, not homogeneous. Janus means hetero means various, and undergo the additional process called the capping, trailing, and where you will see both of them in the eukaryotes DNA, okay, and RNA process making. All right. In the capping, an unusual nucleotide, that is methyl guanosine triphosphate, to the 5 end of the HNRNA. In tailing, polyadenylate tail is added to the 3' end. Means this is a 5' and this is 3'. Here the tailing formed. Means that is this is polyadenylate. Polyadenylate. This is a polyadenylate. This is just for protect the RNA, okay, and also protects the RNA. It's enough, not required. All right, ঠিক আছে? Okay. So the tail is added to 3' template at an independent manner. So we know the template means where it can code. The RNA can be formed. So heterogeneous er moddhe amra etogulo dekhte perechi. Otherwise it was not possible. Next is genetic codon. Genetic codon means the relationship among the amino acids and sequence of the polypeptides. So polypeptides and the amino acids relationship thake, and nucleotide base sequence in the mRNA. mRNA means the messenger RNA which is formed from the DNA itself. Okay, it directs the sequence of the amino acid during the synthesis of protein. Jokhon protein ta form hoy, means mainly they are required in the translation process. এবার কি আমার প্রোটিনটা ফর্ম হবে, কোন প্রোটিন ফর্ম হবে? That is AA. Means the amino acid, there are the 20 amino acids in this. এখানে 20 amino acids থাকে, ঠিক আছে? এই দুনিয়াতে 20 amino acids আছে, সায়েন্টিফিক রিজনে 20 amino acids আছে, যেখানে আমাদের 10টা থাকে ন্যাচারাল and rest of the 10 is artificial. All right. George Gamow suggests that the genetic code should be combined of the 3 nucleotides. So nucleotides 3টে মিলে এর ফর্ম হবে কোড 20 amino acids. অলরেডি সে 20টা amino acids আছে আপাতত কেমিস্ট্রিতে বা বায়োকেমিস্ট্রি রিজনে 20টা আপাতত আছে, যেখানে 10টা হলো ন্যাচারাল, you will get to see in your body, and others are you have to intake them through your diet. Okay, so যেগুলো আমি খাই তো অলরেডি ইজ দেয়ার, যেগুলো খাচ্ছি ওদের ফর্মেশন হচ্ছে, ওদের সিনথেসিস হচ্ছে, আর যেগুলো অলরেডি আমার বডিতে আছে ওগুলো সিনথেসিস, যেগুলো বাইরে থেকে খাচ্ছি ওগুলো তো আমার এনার্জি প্রোভাইড করতেই হবে, আর ওগুলোই যেগুলো বাইরে থেকে খাচ্ছি দ্যাট ইজ কল্ড দ্য এসেনশিয়াল, আর দোজ এমিনো অ্যাসিড প্রেজেন্ট ইন দ্য বডি ইজ কল্ড দ্য নন এসেনশিয়াল. Alright, there are the salient features. See, the code of triplet, 3টে nucleotides মিলে একটা genetic code হবে, 3টে nucleotides. 61 codons are there, amino acids. 3 codons do not code for anything. They are called the stop codon. So stop codon basically UAG, UGA, and UAA. So MCQ may asked. Okay, UAG, UGA, and UAA. So these are the stop codon. Okay. Next, number 2. The codons is ambiguous and specific and code for 1 amino acid. 1 amino acid means 3 nucleotide data form hobe, and a 3 nucleotide er moddhe jai form hobe means it is based totally the RNA related, means uracil will be there, no thymine. Okay, no thymine will be there. Specific thakbe, and it particular codon won't code another one, ja thakbe ektai code thakbe. And this code degenerate, and some amino acids are coded for more than 1 codon. অনেক সময় amino acids গুলো একটা codon বেশিও তারা করে ফেলে, but whatever the codon is forming, it is very specific. Okay, and amino acids are only 20. We have to repeat, ঠিক আছে? But একটা সেম মিলবে না, maybe changes হবে alternative, but same হবে না আর কি. That is what I am trying to say. So this is a tRNA. আমরা তখন mRNA পড়লাম, we already discussed enough, and now it's a tRNA. tRNA means transfer RNA. এর মতলব হলো transfer. অনেক হয়েছে বানানো, এবার আমি ট্রান্সফার করব আরএনএটাকে টু এ সার্টেন রিজন where the genetic code is very important. All right, the tRNA is called the adapted molecules. It has an anticodon. Codon যেমন থাকে তার ঠিক উল্টো দিকে anticodons থাকে. Loop has a basis complementary to the code present on mRNA and also has the amino acid acceptor to which amino acids binds. tRNA is specific for each amino acid. So tRNA is very specific. Another same type of codon cannot be same codon hote pare na. Maybe amino acids can be repeated, but same codon won't be possible enough. Secondary structure of the tRNA is depicted as clove leaf. Its actual structure tRNA is compact molecule which look like an inverter L. So basically this is not L, this is kind of উল্টো এল আর কি। তো এল তো আমরা এভাবে লিখি না, তো এটা উল্টো এল they are showing. As you can see, this is a one amino acid, this is another amino acids. Basically they are the amino acids. Amino acids কে আমরা in short লিখি AN. More further discuss করব about the genetic codon in the next video. Okay, and this is one, this is another one. So as you can see, these are the team RNA, means adapter RNA, and here is a 5 prime to 3 prime. Basically they are the mRNA, that is messenger RNA. All right, so as you can see, this is a codon AGU and এটার উল্টোটা হলো anticodon. এর উল্টো হলো U, basically এর উল্টো থাকে U, G এর উল্টো থাকে C, right? Cytosine. Same ভাবে U এর উল্টো হচ্ছে A, C এর উল্টো হয়েছে G and A এর উল্টো হয়েছে C. And this is how codon and anticodon both are forming, and this is how they are making at the top, that is amino acids. Otherwise same U A U G, এর উল্টো হলো U, U এর উল্টো হলো C, G এর উল্টো হলো A. তো এইভাবে তারা কি বানাচ্ছে, টাইরোসিন বানাচ্ছে, but you can see that the amino acids are different, but same way their codon, genetic codon is also specific. It never be possible, কিন্তু কখনো কখনো ওদের কি হয়, amino acids repeat হয়ে যায়, so that is very normal. All right, but they are close shape, and this is a adapter RNA, this is tRNA. All right, and this is a mRNA. তো RNA basically তিনটা টাইপের থাকে. So RNA, it is required. At that RNA we cannot go through the last process, that is translation. Translation means where you will make the protein itself. যেখানে আমরা প্রোটিনটা বানাবো, এবার আমরা লাস্ট পর্যায়ে এসে পড়েছি। ট্রান্সলেট করব means will synthesize the protein. Okay, all right. Translation process. Translation is the process, the polymerization of the amino acids to form the polypeptides. Polypeptides, the ordered sequence of the amino acids are defined by the sequence of bases mRNA. So mRNA উপর সবগুলো এর পুরোটা mRNA, entire one is a mRNA, and rest of the RNAs are the supplementary, they're not that much, and this is a rRNA, that is ribosomal RNA. Okay, this one, this bigger one, this bigger one. All right, so there are a certain kind of steps. One is a charging of tRNA, here just polar inverted L, as a formation of the peptide bonds between the two charged RNA. The start codon, that is AUG, always remember AUG is a starting one. AUG will start the formation, further formation they will start, and will stop will be done by AAG, UAG, UGA and UAA. Okay, these are the three. And mRNA, some additional sequence that are not translated, these are called the UTR, means untranslated region. কিছু জায়গা থাকে যখন যেটা কখনো ট্রান্সলেটই হয় না, means that is the place is totally restricted, কোনো ট্রান্সলেশন হচ্ছে না, তো ওটাকে বলি UTR. ইটস ভেরি ইম্পর্টেন্ট, অ্যান্ড পুরোটা রিজনের মধ্যে কোনো একটা জায়গা থাকবে যে ফার্দার আর ট্রান্সলেট করছেই না, দে আর নট মেকিং দ্য ফার্দার প্রোটিনস, সো ইট উইল বি স্টপ. For initiation, ribosome binds to the mRNA at the start codon, ribosome moves to the codon to codon, and এভাবে রাইবোজোম এখান ছিল, এখান থেকে এখানে গেল, এখান থেকে এখানে গেল, এখান থেকে এখানে গেল, এখান থেকে এখানে গেল, এভাবে লাফাতে 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 this particular ribosome will be moved from this to this to this, এভাবে এরা চলতে থাকবে, and এইভাবে ওরা প্রোটিনস গুলো বানাতে থাকবে. Series of proteins will form and it will be detached. Series will form, it will be detached. So এইভাবে তারা মালার মতো একটা প্রোটিনস বানাতে থাকবে. Okay, protein chain, which is called a protein chain. At the end, release factor binds to the stop codon. একটা টাইম এসে পড়বে যখন AUG or UAA or UGA, any of them একটা এখানে অ্যাটাচ হয়ে যাবে, and automatically এই যে দুটো বড় বড় রাইবোজোম তোমরা দেখছো, that will be separated from each other. Means this ribosome will be separated with this, আলাদা হয়ে চলে যাবে, and protein যেটা বানানো series of protein chain, it will be separated. Ultimately শেষ, termination হয়ে গেল, and terminating the translation release of polypeptides from ribosome. Okay, so it was the ultimate function of the mRNA, tRNA, rRNA. Three of them are required, and RNA is very important, and RNA formটা হচ্ছে by the help of the transcription and then translation, যেখানে series of protein chain, peptide chain, amino acid chain is forming. The simplest form of the protein is called the amino acids, and যেটা আমাদের বডিতে absorbed হয়. Whatever protein you are eating or consuming, it has to break down into that amino acid. Only then your body cells will be able to absorb the proteins. Otherwise, it's not possible. So the translation, it's important. It's happening in our body cells every time, every second, every moment, to nurture us, to giving us more nutrition. So today's class up to this. In the next video, we'll be talking about the last part of the molecular basis of inheritance, where we'll be talking about lack of parents and other factors of it. Keep studying. Stay tuned with Crazy Science. Thank you.